everyone, and welcome to the 2021 Potato R&D Forum. I'd like to begin today by acknowledging the traditional owners on the land on which we meet today. I would also like to pay my respects to elders past and present. I'd also like to introduce myself, Kristen Sterling, and my colleague, Doris Blazing. Doris, put your hand up. From RM Consulting Group. We are the coordinators of the Australian Potato Pest and Disease R&D program and the organisers of today's forum, which has been funded by the Potato R&D Levy. So during today's forum, you'll hear from a number of speakers who will provide a 10-minute presentation, which will be followed by 10 minutes of Q&A, question and answer time as well. So there will also be at the end of the speaker sessions, there will be a 30-minute panel session, which will involve all the speakers from today from 4 to 4:30. And that will be an opportunity to ask further questions. And we'll also be offering the opportunity to break out into individual rooms with each of the speakers to ask further questions from 4:30 to 5. So that's an optional part of the forum. If you wanted to still know more and ask further questions and have a bit more of a intimate discussion with some of the speakers. So hopefully today's forum should offer you plenty of opportunities to ask all those burning questions. Now, an email was sent out to all registered participants midday yesterday, which contained handouts for reference during the forum. Unfortunately, if you registered yesterday afternoon or this morning, you won't have received that email, but if you contact me after the forum, I can send those handouts to you. The email also contained a guide for participants with just some information about being involved in a Zoom meeting if you're not sure of some of the functionality, and also an agenda for today and tomorrow's session. So if you wanted to know exactly who's speaking when, have a quick look at that agenda. I'll just now hand over to my co-host, Doris.
Hello, everybody, and it's nice to see so many people from around the country from the potato industry. A pity we can't do this face to face, but still. So to make it a bit more personal, if you could actually have your video on, unless the bandwidth, doesn't allow it and you look all fuzzy, but it would be lovely to actually see people. Because we have, and maybe best is to stay on mute, so not everybody can hear the dog barking in the background or the partners talking to you while you're at home. The other thing would be good if you have questions whenever they come to you, to put them into the chat function, which is, if you put your cursor down on the bottom of the screen, there should be a little bubble saying chat. And if you press on that, you'll be able to chat. And everybody can actually read your chat unless you make it secret. So what we will be doing in the discussion, we will use the chat function for questions, because I think we have about 130 people registered, and hopefully most of them will make it. So it's hard to have a discussion. However, as Kristen said, we will have discussion with the speakers individually at the end if you want. So so please, microphones off, faces on, and questions into the chat. We will at the end download the chat, which means any questions we actually haven't answered and which are of interest to everybody, we will answer in an email to you all. So I hope that will be it, and we can get to our seasonal update, Kristen.
Thanks very much, Doris. And hopefully you should have all received a notification when you logged on that we are recording today's session. So there's been a number of people who've been in touch and said they couldn't make it today. So please just note, we will be recording it, and we will be making the recorded session available to those that couldn't make us today. But great that so many of you have been able to make it. So I hope you get a lot out of today's forum. So we thought just to set things off, it would be good to get a bit of an update from around the regions of the season that's been or currently underway in terms of what have been some of the key pest and disease issues. And of course, for some people, it might have been a really great season and there hasn't been anything out of the ordinary, in which case that's great. But I thought we might kick off with WA, beautiful Western Australia. So Sam Taylor, who is an agronomist with Nutrien, is going to give us a very brief overview of the key pest and disease issues for this season. Sam.
Yeah, thank you for that introduction, Kristen. So yeah, very quickly, for conditions south of Perth over here in WA, the Manjimup area continues its usual winter holiday period, I guess, from production, and they haven't really had any unusual or unexpected pest and disease issues over the last season. The typical longer rotation in pasture down that way sort of suggests that they're not going to run into as much pressure, so that's pretty handy for those guys. Coming up towards Busselton, out of season rainfall events sort of through late summer, early autumn caused a lot of botrytis issues through the wine industry, and that actually transferred across onto some isolated potato crops. So that was probably something a bit different that we hadn't experienced before. And when we ran back through some basic fungicide programs, yeah, they hadn't been using products that also cover botrytis. So that caught a couple of guys out. Powdery scab continues to be an issue, but is probably a growing issue, I think, in Busso and also the Mile Up area. So people are looking at their options for managing that. The more recent, traditional winter conditions, particularly through Mile Up, where there's still quite a lot of crop, has been belting things around a bit. So there's been a bit more disease entry for bacterial soft rot and things like that. And outside of unusual pest and disease issues, just probably the growers continually comment about getting quality seed, and there's probably a fair bit to learn in that area, I think. So that's it from WA.
Great, thanks so much, Sam. That's really appreciated. And I would just reiterate, as Doris said, it's great to see everyone's faces, and for the presenters, it is so much better if we can see who's out there. It's quite hard presenting to a whole lot of blank squares. So if your bandwidth allows you to put your video on, please do so. Alan Smith,
yes.
Hi, Alan.
Yeah, good afternoon, everyone.
Thank you, Alan. So I've asked Alan from Snack Brands Australia in Queensland to give the Queensland update. Thanks, Alan.
Rightio, well, I'll try to do it justice. A bit like Western Australia, it does cover a pretty broad geographic area, Queensland. So, planting began in March and is mostly done in the far south and the far north. And I guess the harvesting commenced in June and probably really starts to ramp up now over most of Queensland supply districts. Weather conditions vary understandably across such a big area, but I guess we saw some widespread rain in March in the south, which sort of hit some of our early plant crops and knocked them around a bit, but overall was pretty beneficial generally. So most of the crops are looking really good. And overall, although it's felt like a very cold winter down in the south, it's been quite humid and the soil temps have been surprisingly warm. So we've seen very limited frost damage that we often would see through this time of the year. So out of that, with healthy crops, we've seen no real significant disease. And I suppose the preventative fungicide programs that most people would probably be running have offset any sort of early blight risk from the humidity and the cool conditions. Up in the north, it was they've had a good summer, so there's a good setup for the season up there, I think. But growers have generally reported that it's been really warm, so in fact not really having a winter. And I guess that sort of generates probably a couple of watch outs rather than issues already. I guess we'd probably potentially see the risk of sclerotinia type issues, particularly in the lower altitude areas, probably driven by the weather conditions. It also, the other big watch out there is probably that over on the east here is that it sort of ramps up the reproductive cycles of insects with that have a propensity to chew things. We've heard some sort of limited sort of detections of Heliothis type larvae, but probably the big watch out for us is fall armyworm. And although potato is not its preferred food source, they are aggressive and they have been seen to chew potatoes on when there's nothing else available. And it's probably into its second or third sort of season in Australia. So it's still something very much to watch out for with limited control options. But overall, we've got a fair way to go to complete the season up here. The crops look good. So, yeah, generally a good position.
Thanks, Alan. Sounds like you've made a good head start into the season. I'll just hand over now to Josh Opus. Josh, if you're around, if you wouldn't mind giving the update for Victoria.
Yep.
Thanks.
Can you see me?
I can.
Thanks for that, Kristen. Yeah, look, so looking back at the Victorian season, I suppose we start back in October with our main planting season last year. Over summer, we had a very, very mild summer, a good summer, plenty of rainfall, and it was an extended summer. And one could say that it's probably one of the better years in the last 5 we've seen for both quality and yield. So it was a very good season for on-farm yields and no major disease issues for the processing crop. And that extends, I suppose, into the seed crop as well. So again, very good yields with seed, very good quality, extremely low virus levels and a high rate of certification. So it all bodes well for the planting coming up in October, November, if it does dry out, because we're extremely wet here. But it's the best time of year to get rain in winter. So that's that.
Fantastic. Thanks, Josh. Sounds like Victoria's going well too. We might head over to South Australia. Jack van der Westhausen, if you wouldn't mind giving the update for South Australia.
Hi, thank you, Kristen. Yeah, I can only see what I've seen in my region of South Australia. So this year we've seen above average yields due to the mild weather conditions through the past growing season. Challenges, disease, black dot continues to be the biggest issue affecting the skin finish and the premium packout percentages. It comes to the lack of fresh ground, meaning black dot incidences compounding and increasing year by year. Powdery scab, sounds like some other counts got the same problem, continues to be an issue. There is inferochemistry out there that's shown good results and reducing incidents. So obviously it's good to talk to local agronomists. Verticillium wilt is an increasing problem, also chemistry out there to reduce the incidence. Pest challenges we've seen low incidence on potatoes tuber moth this year. Strips become more and more of an issue. They can be a carrier of tomato spotted wilt virus. Rotation of chemistry to prevent any resistance and to keep the strip numbers under control is what we do. And overall, it was a good season and we've seen some good potato crops. Thank you from South Australia.
Sorry about that. So thanks very much, Jack. Yes, so final lucky last is beautiful Tasmania, and Stuart Millwood, who's with Nutrien, is going to give us the update from Tassie.
Thanks, Kristen. Yeah, I guess our planting season down here with potatoes begins around about September and runs through to late November. And hence our harvest on the back of that begins in January and runs through to about June. We have 2 major processors down here in Simplot and McCain. So the varieties that we grow, Innovator with McCain, and the majority of the tonnes with Simplotter, Russet Burbank and Ranger Russet. I guess we started off with a wet spring, so planting was a bit challenging down here. A lot of our heavier ground, we had to get the ploughs out for the first time in 10 years to help dry that out. But despite that, the efforts of the growers, the crop went in a timely fashion. The growing season was quite a reasonable one. It was what I'd describe as average to solid. Yields with Innovator and Russet Ranger definitely shone in the season that we had, which was quite cool and plenty of rain up to Christmas time. I think that aided in tuber set and the shorter growing season for those varieties. I think the season suited. Because of the cold, wet conditions, especially up to Christmas time, our incidence of powdery scab were probably enhanced. And when I talk about the problem with powdery scab, it's the powdery scab on the roots that our issue is down here with restricting root growth and hence yield. It's not the lesion on the tuber. I guess also Rhizoctonia incidence was up a little bit on the back of those weather conditions as well. And so we did see a bit more greening this year. But overall, like I say, we had a fantastic harvest season down here. We hardly stopped during harvest, so and the quality was overall quite good. I guess our biggest issue that we faced this season, but it has been ongoing, is pink rot. And that certainly not only reduces yield, but also the inefficiencies in harvest caused by that disease is quite extreme. Our size in our crop was probably down this year due to increased tuber set, especially in Russet Burbank. And I guess positives were that we needed a pretty solid season coming off the back of the one before, which it didn't stop raining and we lost a lot of crop. Our solids, our specific gravities were really good. And despite having a really high pressure Irish blight season, we didn't see any incidents of that due to our timely fungicide program. And I guess in relation to target spot, the product that we're using down here, Miravis from Syngenta, is certainly we're getting longer greening in our crops, and we're holding them on for an extra sort of week or 10 days, we feel. So that's been a positive down here. But I guess in summary, pink rot is certainly our major issue down here.
Thanks, Stuart. That's a great overview of what's happening down in Tassie.
Yeah, thanks everybody, and it's quite enlightening. It is enlightening to hear from all corners of the country, and it certainly highlights the differences and, in my view, the importance of having regional research or regional extension, which we hopefully get going in a bigger way soon. So thanks everybody, and it's really good to see what's common and what's different in all industries. So if anybody still has comments to make about their region, please just put it into the chat, because everybody can actually see the chat. Thank you.
Thanks, Doris. Right, well, we might move on to our first speaker for the day. I'd like to introduce Ben Fleay and Greg Hall from Precision Agriculture. Greg, do you want to share your screen?
Hi there. Hello. You hear that okay?
It's a bit faint, Greg. Is it possible to increase your volume?
Yep, just hold on a second.
No?
That's a lot better.
Kristen, is that any better?
No, it sounded
so-
like you.
Do you have a microphone, like a headphone, Greg, that usually improves things? You know, one of the iPhone ones or another one, so you speak directly into the microphone.
Any better? One more. Last one here, is that any better? Is that working?
That's much better. Much better.
Okay, sorry, apologies. Yeah.
All right, if you can share your screen. Ben and Greg are from Precision Agriculture and are going to be talking to us today about management of soil constraints in the potato industry.
All right. Does that screen come up there? Sorry.
Yes.
Yes, now.
There's my start. There we go. So thanks, Kristen, and also Doris for setting up the Potato R&D Forum through OMCG, and also obviously funded through the Hort Innovations funding. It's good to be presenting today. Obviously, a bit of a challenge with COVID, and both Ben and myself are based in well, I was going to say sunny Ballarat, but not so sunny Ballarat out the window at the moment. And we're going to be talking today about you know, what precision agriculture has been doing around managing soil constraints. So I guess Precision Ag is as a business that's been around since 2009. It started really in Ballarat and the Western Districts and Southern New South Wales and the broadacre regions, looking at you know, identifying soil constraints and helping growers, you know, improve their yields mostly through some sort of zoning, through mapping or yield analysis. So as the business grew and got bigger, we got more into grid-based soil sampling, and that's I guess seen our business grow to around 30 staff around South Australia, New South Wales and Vic. We also have some regional offices in both Queensland and Tasmania. Got quite a large R&D team, and I guess the drive for our business is to produce grower outcomes. So we're not just you know, producing a nice map. We actually want to get an outcome out of it. So this year we'll probably see us producing testing in excess of 200,000 hectares, and that, you know, really makes us probably the largest independent soil testing business within Australia. I started with Precision Ag last year as the National Horticultural Sales Manager, and as part of that, the remit was really to take what we'd learned in broadacre and bring it into horticulture. So my background was 15 years in the seed business, mostly based in Tasmania. And as an aside, I have I guess a farming enterprise in Tasmania that grows seed potatoes for both McCain's and Simplon. So I guess from a personal point of view, I've got a strong interest in the potato industry, and it's nice to see quite a few people there today who are familiar with. So I guess when I'm thinking about soil sampling, I was thinking about potato production and some of the major constraints, really thinking about nutrient and water. And if we're thinking about nutrients, what Precision Agriculture has been doing is this grid-based soil sampling. So for those of you that don't know what a grid-based soil sample is, instead of just taking one soil sample in a paddock, we break the paddock down into sort of smaller grids. And this might be, you know, a 1 hectare grid size, for example. So a 10 hectare paddock will have 10 grids. And then within each of those grids, we will take 8 subsoil samples. We use a little motorbike based sample with a GPS unit on it, and it takes a core sample and then put it into a bag with its own barcode and its own GPS reference. So when we send it to the lab to get analysed, we can then take that back to that exact spot in the paddock. So I guess why worry about it? Well, in horticulture, you know, uniformity is ultimately the king. And I know this is not a photo of a potato paddock, but it's quite a nice photo, just showing why uniformity is important. And it doesn't matter whether you're growing, you know, seed or ware or processing potatoes. there's really quite a nice sweet spot for making money. And yes, you get paid on yield, but the yield's got to be within that spec range. So what do we, what can we do to, to give ourselves the best chance of being uniform? We can start by growing it in a uniform media. Now this is an example of a grid soil sample. So this is a pivot, it's actually South Australia. And these are 1 hectare grid sizes. And for this example, we're looking at the coal pea. So within this pivot, we have a coal pea ranging from a low of 7, sort of in the middle there, going right up to 76. So there's quite a large range within one pivot to try and grow a really uniform crop. And we know that pea is obviously important for things like seed set. The more pea that's available, generally, the better the set. The better the set, obviously the better the yield, especially if that's uniform. So what can we sort of do to, I guess, give ourselves the best chance of making that soil uniform? If we take those samples and we put it into our solely platform, we had a discussion with this grower in particular, his agronomist, and they wanted to aim to have a coal pea of 65 across the whole pivot. So we put that into our platform, and then using MAP as the base for bringing up the pea rates, we can increase or add FERT more specifically to the lower areas that need more, and then reduce it on the areas that need less. So we've got ranges there from 600 kgs per hectare all the way down to nothing and ever in between. So we've put 11.2 tonnes of MAP on that paddock, but we've really targeted the different areas to try and increase or decrease, well, not so much decrease, but certainly increase the P level so it's more uniform to start with. Now, obviously, we can do this across any nutrient. So, for example, we can do potassium. In this example's just exchangeable potassium. You can see highs of 281 all the way down to I think 90 is about the lowest there, or 83. So again, what this grower decided to do was use a base rate across the whole pivot, and they using SOP, putting on 250 kgs per hectare. But then in the lower areas, just try and increase those potash levels by putting some extra on. So in this case, they put 25% extra on, which works out about another 60 kgs per hectare to try and increase those lower areas. So we're really trying to give ourselves the best chance of making that medium uniform so that we can get a uniform product. Now we can do this on any grid size, we can do it on any nutrient. So I guess the keys for us really for potatoes we're looking at currently are pH, P, K, magnesium and calcium, but of course we can do traces, nitrogen, carbon, organic matter, anything that's really specified by the grower. I think as we get more into this and get into other areas outside of yield and uniformity, whether it's disease or different root diseases or leaf diseases, we're going to see a lot more interest in other nutrients to increase the nutrient levels across the pivot and make them more uniform. How we go for time there, Doris?
You've got 2.5 minutes left.
Brilliant. All right, the other way, if we're thinking about managing water, is obviously managing soil constraints through water. Now, we can overwater or underwater, and that can have a huge influence on how our crops go. So one method is to obviously take what's called an EM38 map, which really just identifies the textures or the different textures within the soil. And so if we take an EM map, for example, on this pivot, we can identify some lighter areas, which are the red areas, and then in between some heavier areas, which are the blues and the greens. And then we can use that to make a variable rate irrigation map. Now, this is not exactly new technology, but it's been quite slow to be rolled out within the potato industry. And part of that is around, I think, obviously, I guess, skepticism about how reliable perhaps some of the technology is, and just how complicated it is to implement along with the cost. But just to improve the, I guess, the water savings or the applications of water across the pivot can actually influence quite enormously the amount of returns back to the grower. We can also use those same maps to develop drainage maps. Now, you know, we take the elevation and the EM data and develop a water flow map, and then use software to put these drains within the pivot, which is the pivot on the right, these blue drains, and make a simulation to show what rainfall does. So we can actually influence the size of the drain or the slope of the drain, or just the cut of it to remove more or less water, depending on what we're trying to achieve. The last thing I'll talk about just really quickly, we've been playing around with this a little bit of Tasmania, was using variable rate for planting of seeds. So this is a photo of Delarame with a quite nice uniform soil. Then what we're looking at doing is perhaps adjusting on the go the seeding rate, which is quite common in broadacre cropping, and they're now doing 4 or 5 different adjustments on the go whilst they're planting, whether it's seed or different fertiliser or different nutrients. So this example is just a dogmac planter with a Topcon controller in it, so we can adjust in the cab the seeding rate or the fertility rate or the Amstar. Now we can actually do that on the run, and what we're looking at doing is developing an EM map or an EM38 map. So in this example, the top dark red by the solids at the very top there was quite a dry area, was sort of lighter stony area, and the spuds were quite small in that area. And then when you come down into the darker blue areas, it's a lot heavier, and the spuds are quite large. So what we're looking at doing is to change the density of the spuds while we're driving along. So in those red areas, we were widening the set, the distance between the sets to give it a better chance of making size, and then in the darker areas, tightening it up to make it more competitive within the row. So we're hoping to try and make a more uniform product across the board.
Thank you very much, Ben and Greg.
Thank
Is there
you very
anything you
much.
wanted to finish up with?
I'll just, I guess if you look at, perhaps the losses of some of these things, if you only have to consider, say, the price of processing potatoes this year at around $350 a tonne, maybe it's a fraction more, a 60-tonne crop, that's $21,000 a hectare. If you lose, 1 hectare in a 50-hectare pivot, that soon adds up to a lot of money. So some of this technology can be used, I guess, over a wide period of time, not just on potatoes but other crops. So when you spread that, that small sort of upfront cost across a range of crops under a pivot or under other forms of irrigation, it's quite cheap.
Thanks very much, Greg. Look, we'll open up the floor for questions now to Ben and Greg. If you have a question, can you please type it into the chat function, which you'll see there's a little chat button at the bottom of your screen. If you click on that, it'll bring up a chat board, and you can just type a question in there. And Doris and I will ask those questions of Ben and Greg, but thank you very much. Doris, did you have any questions?
Yes, I mean, while we're waiting for everybody to get their questions down, Greg, I was wondering about a couple of things. That with the pivot you showed, it looked as if there has been nearly a row crop before, like an orchard or something. Do you know what was in the paddock previously?
No, I think it was just the way, that was actually South Australian pivot, so it was probably just the way the sand is either blown or being shifted.
Yeah.
And moved within the pivot, which obviously you see quite a lot of in South Australia.
Yes, it's probably like a sand dune country.
In the sort of swales.
Yep.
From east to west, I guess.
Yeah, that's quite interesting. And another question I had is, you do the grid sampling. Would it also make sense to do more, like looking at the Delarame side, do more targeted sampling where you actually sample the areas where you have different yield or can see different NDVI images?
Yeah, so I guess if you look at broadacre as a proxy, maybe originally that was, I guess, the theory was to break the paddock down into zones using an EM map and then really specifically target some of those areas. And that's still quite common as a lower cost way of looking at your soil constraints. So you might, for example, take 2 or 3 soil samples within a zone, but overall take a lot less soil samples. The problem with that being that we haven't really been able to see a great correlation overall when you look at it compared to a grid-based soil sampling across the whole paddock. It's better, but it's probably still not quite as accurate as a grid-based soil sampling. But I think, Doris, I'll say there's some merit in that on certain things, especially root diseases or some other disease that's really influenced perhaps by the soil texture or the water held within that profile of that particular zone, whether we are looking at pink rot or powdery scab maybe, or carrot type problems that come about from overwatering, underwatering, or those different zones. I think that's got a lot of merit.
So Ben, here's a question from Jenny Ekman, so asking about the cost of the grid-based sampling, and also, does it make economic sense?
What's the rate of return roughly?
So I think I'll just give you an average price. So to do a 1 hectare grid sample on pH, P, K, Mg, calcium, and sodium. That's a fairly standard test for us. That works at about $65 to $70 a hectare. We generally see that saving. It's hard to be specific, but I generally would see that saving just in the line rates. So we might say that cost of that doing that on one pivot is paid for almost by the savings in line. And then everything else on top of that, whether it be savings in P or K, and not counting production savings or increases, pays for the cost of doing it.
So do you actually get or have evidence of more even yield, like even more, once you adopted either pH or even out the fertilizer rates?
Well, I guess we're still a little early days, Doris, particularly potatoes, but COVID didn't help us for travelling around some of the sites we've had for this year, which is a bit disappointing. But certainly we're just waiting to see some of the pack out information from the fresh market stuff we've done in SA, and also just some of the data we've got from Tassie. But it certainly looks like the uniformity is increasing, but there's quite a bit of ground truthing to work out more specifically, I think, around what are particular nutrients and how they interact. And that's really different, say, in SA compared to Tassie, compared to Victoria. And it's really got to be more driven by the agronomist or by the grower themselves. So I think, for us moving forward, we've got to work more closely with the agros within the business to try and understand just how those constraints matter.
So there was a question from a few people about adoption rights. Are people, ringing you every day wanting to get data for potatoes and look at drainage or variable rate, or is it still you chasing the market?
About 50-50, I think, at the moment. So certainly some of the bigger players, we've done quite a lot of work in already the last 2 or 3 years, and then I guess this year we're probably just starting to see the seeds of that come to fruition, whereby we're getting some references now and growers ringing us. So it's still early days, but certainly it looks like, and not just potatoes, I guess onions, carrots, and some of the tree crops, we're starting to see some referrals coming through to do more work.
And have you done some work on biological indicators like general soil biology or mycorrhiza, or also any disease DNA testing?
So not specifically potatoes yet. Certainly we've done stuff with club roots. We're sort of working towards putting together a project maybe to do how club root is obviously affected by pH. So in brassicas, that's one thing we're working with. Sclerotinia in lettuce is another one we've been working with one of the seed companies on to kind of monitor how these mustard seed crops or biological crops can decrease the rates of sclerotinia within the soil. And that's probably, I think, an interesting one, certainly potatoes, whereby maybe we can measure whether it has an effect on, say, powdery scab or pink rot or some other diseases, these mustard type crops.
We also get a request here from Michael Trenter for you guys.
Actually, Doris, we might just finish, sorry, we might just finish it off there. Apologies. So thank you so much, Ben and Greg, for that presentation and discussion. And as we go on, there will be a panel session at the end in the breakout room. So if you had some more burning questions for Ben and Greg, you'll get an opportunity to talk to them in more detail a bit later on. So we might now move to, I'd like to welcome Dr. Sally Anderson, who has joined us from beautiful New Zealand. And she's going to be talking to us today about Tamarixia. Sally, are you there?
I am, Kirsten.
Great. I'll just get you to share your screen.
Yeah, sure.
So Sally works with Market Access Solutionz, and she's the Scientific Services Manager for Market Access Solutionz, and designs, manages and implements research programs on behalf of horticultural industry clients in New Zealand. And yes, she's going to talk to us all about Tamarixia. Take it away, Sally. Thank you.
So that presentation's up, Kirsten?
Yes, it is.
Oh, cool. Okay, hi everyone. Yes, as Kirsten said, I'm over here in Wellington in New Zealand, where we're happily COVID-free at the moment. So apologies to those of you that are in lockdown. Yeah, so today I'm going to be talking to you briefly about a project that we've had running here in New Zealand for 3 years. It's a program looking at Tamarixia triozae as a parasitoid of tomato potato psyllid. This research program was funded by a number of vegetable product groups and industry support, and also our Ministry for Primary Industries, who is a government co-funder in this program. We also had Plant and Food Research as our science research provider. So just to recap on the New Zealand story of psyllid, I'm not sure how many of you are familiar with this, but I'll just give you a brief overview. We first detected tomato potato psyllid in New Zealand in 2006. When some surveys were done following that first detection, it was found it was actually quite widespread. So it had spread beyond the tomato greenhouse. It was into potato crops. At that stage, it was decided it was too far spread to be eradicated. In 2008, the news got even worse in New Zealand. We detected Candidatus liberibacter solens serum, or LSO, and this is the bacteria that causes zebra chipping in potatoes. Surveying psyllid at the time, we found LSO was present in psyllid, so we had the bacteria and the vector, not a great combination, and so that's what we call hot psyllid. So psyllid that's going to be vectoring the bacteria. At that stage, IPM programs were lost. Growers had to revert to calendar spraying, particularly within tomatoes and potatoes. Since that time, things have actually improved a lot for growers in New Zealand. They've managed to get a better handle on how psyllid moves around, seasonal activities, and there's been a lot more chemistry come through for control of psyllid as well. So a lot more awareness on how, from the growers' part, on how psyllid, how to better manage psyllid. From 2009, we've been researching solutions through a number of government and industry co-funded programs, and the program I'm going to talk to you about today is the Tamarixia release program. So Tamarixia triozae is a parasitoid and a predator of psyllid. The female lays a single egg on the underside of the psyllid nymph, and the females prefer 4th and 5th instar psyllid. And a single female can lay up to 165 eggs during its lifetime, and its lifetime lifespan, sorry, averages about 20 days. So what happens is that the Tamarixia lays the egg, the larva develops within the psyllid nymph. So you can see in the photos to the right, you can see that larva developing, and eventually that larva will develop into an adult, and the adult will chew through the psyllid husk, and it will basically escape from the husk of the psyllid. It takes about 12 days from egg to adult emergence. And the other, I guess the other benefit of Tamarixia is that it's also a predator. It's not just a parasitoid, it will predate TPP. And females use the ovipositor to puncture the TPP nymph, and then they feed on that hemolymph. And that hemolymph provides a nutrition source, a protein source for egg formation as well. So just in terms of overall mortality, that Tamarixia, the effect is around 70% is parasitism and 30% is by predation. So just in terms of the release program, the objective was quite simple. It was to establish self-sustaining populations in New Zealand. There were two aspects to the program. There were the grower-led releases, and there were the plant and food research releases. And they were specifically to study survival, the levels of parasitism, and how far Tamarixia might spread. These releases occurred for 3 years. It was a 3-year funded research program. We started that program in 2017, and we completed it in 2020. I'd have to say before the program, we had to go through quite a few hoops with our New Zealand EPA to obtain a release approval. Tamarixia is an exotic biological control, and New Zealand has a very strict regulation around importing and releasing exotic insects and particular biological controls. We got that approval in 2016, and then we imported Tamarixia from Mexico, and there was a bit of quarantine screening, but then we began rearing for release. So just to give you a bit of an overview of what the release program looked like, it was pretty modest in year one. We only released just under 2,000 Tamarixia. We didn't really have a big rearing program up and underway. We got ourselves a little bit more organised, I guess, in year two, and we released just under 7,500 Tamarixia. In year three, which was the final year of the release program, we basically went all out, and we managed to release over 20,000 Tamarixia. One of the really encouraging aspects of the program was not only the grower releases and the interest shown by growers, but also home gardeners. They were really keen to take up the opportunity to release Tamarixia, and we had over 10,000 Tamarixia released by home gardeners that year. So this is just really a series of photos to give you an idea what the rearing conditions look like. So Tamarixia is an obligate parasitoid. It needs a TPP host to be able to complete its life cycle. So you need to rear TPP on host plants. So in this case, on the left-hand side image is insect cages with tomatoes in them. And then once the TPP, the Tamarixia have hatched, you can collect the adult Tamarixia. These are just simply, sorted into vials, and then the vials can be sent at room temperature to a grower or to a home gardener and to release into a crop. So the bottom right-hand side of the screen is just a vial that's had its cap off. It's very passive. It's been released into a potato crop. Alongside the program, we had plant and food research doing their own releases, and they were really focused on releasing into non-crop hosts. So the reason for this is that non-crop hosts in the landscape are essentially a source of psyllid, and that's where psyllid basically come from into the crops that you plant every year. So they were releasing Tamarixia into these non-crop hosts. In particular, it was African box thorn, and also to our native porroporo, which is a species here in New Zealand. And you can see the box thorn on that bottom right photo can get pretty big. So you can see the scale of that hedge, and just imagine how many psyllid could be hiding out in that hedge. So yeah, so we were looking at can we release Tamarixia? Will it survive in those environments that we're releasing it into? And will it survive for any length of time? So that question was addressed by Plant and Food by looking at overwintering survival. So the process was release them in the first year and then go back to those same sites and see if we can find Tamarixia. And amazingly enough, given the small numbers that we released in that first year, we found that Tamarixia were present at quite a few of those release sites. So 4 out of those 6 release sites, we detected Tamarixia adults, which was a fantastic outcome. So the big tick there was that Tamarixia will survive over winter. The other question was how effective as a parasitoid is Tamarixia? So that infested host plant material was gathered up and taken back into the lab, and the incubated for a period of time. And then what you can do to estimate levels of parasitism is actually look for psyllid nymphs that have that exit hole in them. So between 4 and 40% was the estimation of levels of parasitism, and an average of 15% parasitism. So that really is another big tick for this biological control. So just looking at spread, that was the other question we wanted to look at. We released at several sites within the Hawke's Bay and within Canterbury, and then plant and food scientists went around and looked at other non-crop hosts to see if they could find Tamarixia. And amazingly enough, in the Hawke's Bay, we found Tamarixia at a site 24 kilometres away from the original release site, which was fantastic. Not quite as effective, I guess, in Canterbury. It's probably because the landscape is a little bit more patchy, and there's less crop and non-crop hosts within that Canterbury area. But still, the fact that Tamarixia was found away from those original release sites was a big plus for us. So just in terms of resources that we developed for growers, and I believe that these have been loaded by Kristen and they're downloadable from that forum invitation. So please feel free to download those resources and have a look. Yeah, so this information was provided so that growers could get an idea of how to release Tamarixia, how you follow up and do actual surveys on the ground, and some considerations around insecticide use when using biological controls. So anyway, just in summary, the 3-year program, we released nearly 30,000 Tamarixia by growers and home gardeners. The releases and surveys that we carried out over those 3 years have shown that Tamarixia survives quite well under New Zealand conditions. Parasitism rates were detected up to 40%. The spread up to 24 kilometres from a release site in the Hawke's Bay. So our conclusions were that yes, Tamarixia could be and is an efficient biological control agent of TPP. So I guess that, there'll be a lot of questions out there. What does it mean for Australian potato growers? Well, it is a viable biological control option. It's a specific psyllid parasitoid. It's not a generalist predator, but it will work alongside naturally occurring predators. But you do have to consider appropriate use of crop protection tools if you're considering using biological control agents. So what I mean by that is you need to use softer chemistry. You need to think about when you're using that chemistry as well, and you need to do your field scouting and your monitoring. In New Zealand, through the program, we've actually now have a commercial supplier of Tamarixia. So growers can actually purchase Tamarixia and release. And as I said, home gardeners are also taking up that opportunity, which has been great. And a really unexpected outcome of the program, but it's something that we really celebrate here. that's me. Yeah, so really pleased to be able to share this information with you, and I just welcome any questions.
Thank you, Sally.
Thanks so much. Sorry, Doris. Thanks so much, Sally. Yeah, we want to stop sharing your screen. So the questions have already started flooding in. I don't know, Doris, do you want to ask some of those?
Yes,
Sally?
I mean, there was really obviously great excitement to be prepared in Australia, or to have it at least in WA. I would think we need to go through similar or worse hoops here with the EPI or with also the APVMA maybe. Does anybody have any information on activities in Australia to get Tamarixia into the country, or the viability? If we might make a exception for somebody to talk if you put your hand up, because I'm not sure that we're actually doing anything. Sally, do you know of anything?
Sally,
I'm not aware, no.
How long did it take to get it approval to use in New Zealand?
From initiating a research program to the actual approval, which was in 2016, it probably took us a good 4 years.
Oh.
Because the reason I say that is that we actually had to do a lot of host range testing here in New Zealand we have quite a considerable New Zealand native psyllid fauna. And so we had to prove to the EPA that it wouldn't actually have a detrimental effect on that native psyllid fauna. So there was quite a considerable amount of host range testing that we had to do to prove that. There's also, we had to conduct an economic assessment and a risk benefit analysis. It's all part of the way the EPA here in New Zealand works. I think in Australia, there's a similar process that you have to go through. You certainly do have to do some host range testing. You would have to do some host range testing to prove that you wouldn't have a detrimental effect on your native fauna. You would also have to do consider how conduct a risk analysis as well, I think. Unfortunately, I'm not really familiar with the Australian situation, but certainly in New Zealand, it's quite a considerable process to get an exotic BCA approved.
It would be here too, given the the number of native psyllids we have, and also, I mean, there's a question there about: did you find that Tamarixia did attack any other beneficial insects?
In the lab, no choice testing, yes, we did. There was one native psyllid, I can't recall the name of it, but it wasn't a preferred host. So there's always the argument that when you're doing lab testing on host range testing, that it's quite artificial, and so you need to consider what will happen within the lab is not necessarily reflective of what will happen in the environment, and that's always been a bit of a contradiction when you're trying to analyze those host range testing results. But I guess the short answer is that there wasn't enough evidence to support that Tamarixia would parasitize native psyllids in New Zealand.
There might be an idea in our questionnaire on the end of today, if people who are interested in any activity, about Tamarixia in Australia, to actually note that on the survey, so we have a bit of evidence for whoever wants to follow it up, that people really in the industry ask for it. Yeah, so there's actually a question about, should we initiate a research project? So that was the thing. If we can only initiate research project these days with board innovation if enough people ask for it. So please do, because I believe we will at least have 4 years research to do.
Yeah, and Sally, was the project driven by Potatoes New Zealand, or who sort of initiated the work?
Yeah, initially it was, it's always been a collective. Potatoes New Zealand, Tomatoes New Zealand, New Zealand Tamarillo growers, all the sectors, all the vegetable sectors that are affected by psyllid. And obviously potatoes is the biggest sized industry in New Zealand, so they carried the bulk of the work.
All right, well, we might finish there. Thank you so much, Sally, for your presentation. That was really interesting. And look, I know there's been a huge amount of interest in TBP and its potential impact in Australia. So we're always keen to see how New Zealand have done things in regard to TPP. We might break for 10 minutes to have a bit of a toilet break, a cup of tea break. We will start again promptly at 3:10. So everyone has 10 minutes to shoot off and get some refreshments, and we'll see you back at 3:10. Welcome back, everyone. We'll get started again. Hope you've all had a minute to refresh yourselves. So I'd like to start off with a video that was developed by GRDC, the Grains and Research and Development Corporation. It features Professor Jean Ristaino of the Emerging Plant Disease Cluster at North Carolina State University. The cluster has created 2 smartphone-based sensors that interpret plant volatile organic compounds and DNA to identify disease in the paddock. And one of the diseases that they've been working on is late blight in potatoes. So this new technology could potentially change the way Australian growers identify and respond to crop disease outbreaks.
Perth Grange Research Update. Growers and advisors got a sneak peek at the latest plant disease diagnostic technology being developed in the US.
So we're very interested in new diagnostics that are quicker and that can detect disease prior to symptom development in the field.
Professor Jean Ristaino directs the Emerging Plant Disease Cluster at North Carolina State University. Alongside engineering collaborator Dr. Xingshang Wei, Jean and the research team have created new technology that can identify crop disease in the paddock via a smartphone.
Generally, when diseases are identified, you have to go out in the field, get a sample, bring it into the laboratory, either isolate the microbe in culture or do a DNA analysis, which can take days to weeks for a diagnosis. So the technology that we're developing is more rapid, and it can be deployed in the field, and it's really a simple technology that anyone could use.
There are 2 different in-field sensors. One analyzes volatile organic compounds, VOCs, while the other analyzes DNA. Both technologies have been proven to accurately identify disease before symptoms have occurred.
Well, when a plant becomes infected by a pathogen or an insect pest, it releases a series of volatile organic compounds, and these volatile compounds are signatures of infection. So what we figured out with some fungal pathogens is we can inoculate a plant and before symptoms occur, within 2 days after inoculation, the plant starts to emit the volatiles. We can read the volatile signature and identify the pathogen before symptoms.
The VOC sensor works by placing a leaf in a small container for about 15 minutes. A device attached to the smartphone then sucks air into the chamber that contains a paper strip. The strip is embedded with an array of chemical reagents that change colour when they come into contact with a specific chemical group. After just 1 minute, the strip will be able to indicate which disease is present. And it's proven to be 98% accurate.
Microneedle technology is a small patch.
The second sensor uses plant and pathogen DNA to identify disease. It's called microneedle patch technology.
Microneedle patch technology has been used for a number of years now in the medical field. If you know someone that has diabetes or is trying to stop smoking, you may have seen a patch being used. They're used to deliver drugs in humans, but they also can be used to gather information like blood sugar levels, etc. So we're taking this microneedle technology and applying it to plants so that we can actually measure the health of the plant, measure the DNA content, or we can measure what pathogen is there. So the microneedle technology is kind of a new technology that will allow us to monitor plant health.
So how's it work? Well, a microneedle patch is attached to the leaf of a suspect plant, held in place for a few seconds, and then peeled off. It's then rinsed with a buffer solution to wash genetic material off the microneedles into a sterile container. From there, specific assays are run to identify what disease is present.
We're running LAMP assays, which are isothermal, very rapid, quick assays for identification of pathogens. And many crops aren't infected by one pathogen, they might have multiple pathogens. So by tailoring the cocktail of assays in our LAMP assay, we can multiplex and identify multiple pathogens at the same time from the DNA. We remove the DNA from the patch within about a minute after puncturing the leaf. A typical DNA extraction can take hours or even days, depending on how elaborate you get, using expensive kits. These patches cost cents to produce, and we can make them out of biodegradable materials as well, so that we won't have to deal with plastic.
So far, the team at NC State has focused on using these sensors to detect key fungal pathogens for the potato and tomato industries. But with such promising results, the scope of the project is expanding into bacterial and viral diseases, as well as more crop types.
Well, we started out the work with Phytophthora infestans. It's the pathogen that caused the Irish famine. It infects potato and tomato, and it results in billion-dollar losses globally. And we're planning on scaling the technology into the field and looking at multiple pathogens as they occur in space and time.
Sensor technology that can be attached and left on plants is also being explored, meaning growers could be immediately alerted to pathogens, even if they're not physically standing in the paddock.
Wearable sensors might allow us to monitor plant health over time, but they have to be resilient to climate, and ideally we want to wirelessly transmit data and not have to physically go out and remove the patches or the volatile sensors. So these are the kinds of things we're working on now, deploying in the field, because all of our work's been done in Phytotron in a plant growth chamber, and we haven't deployed them in the field yet. So that's really the next big step, to know if they're going to be field ready.
With a lot of commercial interest around these two sensors, Jean's optimistic about the future of this technology and its adoption across the grains industry.
So we have a lot of opportunities in future directions we can go with this technology. The more I read outside of the discipline of plant pathology and traditional diagnostics and read more into the work that's going on in the medical community, I'm seeing a lot of opportunities for new applications of this technology in agriculture. Growers are spending more money on technology and data analytics. Crop consultants are using this data to monitor farms over space and time. And I can see this technology fitting in nicely into crop monitoring platforms that growers are already starting to buy into.
Professor Jean Ristaino, plant pathologist at North Carolina State University. And this video is one in a series of update videos recorded at the 2020 GRDC Grains Research Updates. Go.
So I just thought that video would be of interest. Jean was obviously invited to talk at the GRDC R&D updates last year, and as she mentioned in that presentation, a lot of the field work, a lot of the work that they've done with that technology has been done under controlled conditions in growth chambers, and they haven't done a lot of work in the field yet. My understanding is that it's not something that's being done in the potato industry at the moment. I know that, for example, in the nursery industry and for quarantine, that they do use LAMP assays, so that style of technology to screen for pathogens. Doris, did you have anything you wanted to add on that one?
I'm just actually posted a message again in your survey. Please, if you're interested, this is long-term blue sky technology, but we need to start to get it eventually. Also, good news is that there is similar technology it's probably even more ready in Australia to look at soil health and to actually do in-field testing using iPhone apps on soil health. So watch this space. We keep you informed as long as our project lasts, which is just until the end of next month. but also through the soil wealth and integrated crop protection project, which has a slightly longer lifetime. And I'm sure AUSVEG will also, and the extension project together will let you know of any new developments.
Thanks, Doris. So we might move on to our next speaker. And again, if there's any particular questions or discussion that people want to have on that video that we just shown, we'll have a bit more time in the panel session to have a chat about that. But I'll move on to our next two speakers, which are Callum Fletcher and Shakira Johnson from AUSVEG. Shakira will be talking first. Shakira, if you want to start sharing your screen. That would be great. I'll just right onto it. So Shakira will be talking about iMapPESTS and Callum will be talking about emerging insect threats to the Australian potato industry. Callum is the biosecurity coordinator at AUSVEG, and Shakira coordinates the communication engagement activities for iMapPESTS, which she will tell us all about now. Thanks, Shakira.
Great, thanks, Kristen. Hi, everyone. You can hear me well? Yes, great. Okay, cool. So this is just a quick introduction and overview for those of you that don't know about iMapPESTS and those of you that are a little bit familiar with it. So just, there we go. So the iMapPESTS project is a project out of DOR's Rural R&D for Profit program, and it's led by Hort Innovation with support from each of the plant industry RDCs across Australia. It's made up of several different research sub-projects that are looking into surveillance and diagnostics technologies. And then those research outcomes are translated out to industry through comms and extension activities by AUSVEG. It's basically a national program of research, development and extension designed to put actionable information into the hands of Australia's primary producers, so potato industry, to enhance on-farm pest management decision-making. So it starts with our smart surveillance technology and is underpinned by cutting-edge pest and disease diagnostics. And then all of the data that we're gathering from those activities is tied together in a customized data collection management system and then pushed out through engagement and adoption activities. And that's all with the aim to enhance pest management and improve biosecurity and area freedom outcomes for industry. So the surveillance side of things, we have a couple of different projects looking into the surveillance. We've got SARDI doing these mobile plant pest surveillance units, and then WA's DPIRD looking at trap networks across the grains belt in WA. And some of their work has application across other industries as well. But I'll focus on SARDI's Sentinels for now. So SARDI have been focusing on the development and construction of these mobile surveillance units, and we call them Sentinels. Some of you may have already seen some of these units. You may be familiar with this one in particular that's up on your screen now, that's Sentinel-1, but we currently have 6 of these Sentinels and they're all a little bit different from each other. And we are currently building Sentinel-7, which is different altogether again. The trailer units or the boxes, the Sentinel units, have specialised trapping equipment on board, and they feature traps that are designed to sample airborne spores or insects. Here we have our 6-metre insect suction trap, and the yellow cap trap here is for the 2-metre insect suction trap. We also have an onboard weather station that collects environmental data at the time of sampling so that we can tie it all together to make sense of what's going on. And I know, Jack, you mentioned earlier, thrips were a major problem in South Australia last season. And I just thought I had some other graphs in here, but this is an example of the outputs from the Sentinel surveillance activities. And this was from a short period of surveillance activity that was going on out at Virginia with our Sentinel-4. Now you can see here, this is our environmental data. We're collecting temperature, rainfall, and humidity, weather, and I just wanted to show you an example of the kind of thrips numbers we were picking up last year. So you can see massive peaks of western flower thrips and onion thrips, and up in the Barossa we were seeing around 3,500 thrips in some of those peaks, which was quite surprising because we weren't really thinking we were going to see thrips that far north in that kind of region. Now, all of this information about the trials, the different trials we have across Australia, is all available on our website. It's kind of a hub for all of the information. If you want to look at the different types of sentinels, you can see it here. You can get in contact with us through here. You can look at previous trials. So you can look at that Virginia trial. We've done heaps of trials around South Australia, thanks to COVID, but we are now getting out across the country into other regions. So we currently have sentinels in Lockyer Valley, that's Sentinel 6, and Sentinel 5, which has our new style 6-metre trap. That's at the Tamworth Research Centre. Now that's focused on broadacre, but we are also monitoring for horticulture, pests and pathogens while we're there. Next up, we're deploying Sentinel 4 into the Wimmera, again looking at broadacre, but what I hear from a lot of agronomists down in the peri-urban region is that they look to broadacre for a bit of a heads up about what might be coming their way after broadacre harvest. We're also keen to get back into Virginia and work more closely with potatoes and see if we can monitor and work closely with those industries to overcome some of the issues they're experiencing. And we want to go back to Hart Field site, which is another broadacre site, to do our 3rd year of trialing there, so we can have some comparisons between years and what we're picking up, which will be really interesting because we're starting to see some differences. We're starting to see earlier occurrences of those aphids and thrips through our landscape, and that can have important impacts on industry. We're heading up to Cairns after the Lockyer Valley in a sugarcane trial, and then Sentinel 5's heading down to Griffith. And a nod to Jonathan, who's in the audience here, we are going to Sydney Basin, working with local land services. That will probably be Sentinel 7. Sentinel 7 is going to be a completely different type of sentinel, which you can sort of plug in and take off different samplers, and you can put them down on the ground, you can put them on the other side of the paddock, and you can do a lot of spatial analysis of movement of pathogens and insects across that crop. So these two are quite important for potatoes, I would say, and what I'm really interested in those trials is looking at the insects as vectors of viruses and the incidence of insecticide resistance in those populations that we're picking up, because that's going to make it harder to manage these pests. And then of course we've got the benefit of early detection of pests such as TPP and also the leaf miner. We now have American leaf miner and serpentine leaf miner in Australia. So I'm happy to say that we'll be able to report as soon as we can on those. That's it from me for now, but please get in contact and Callum will take over from here. Thanks.
Thanks so much, Shakira. We might just hold off any questions for Shakira till the end. Callum.
Brilliant. Thank you. Can you hear me fine? Hi, everyone. My name's Callum Fletcher. I'm the Biosecurity Coordinator at AUSVEG. And look, I think what I'm just going to talk today about is my work is basically focused on anytime something new comes into the country that affects the vegetables or potatoes industry. I'm integral in the process by which the decisions to make and manage or respond or eradicate are made with state and federal government and industry levels. Now, this past year or year and a half has been a pretty busy year in the potato space. So I was just going to talk about 3 quick examples. Alan has already mentioned with fall armyworm. Shaq has already mentioned with the various leaf miners. And that Sally has actually already mentioned with the tomato potato psyllid, but I think we've got a little bit better news on the potato psyllid, so that's good. So every year we do a review of the high priority pests through Plant Health Australia and the federal government. Currently, the ones that are high priorities are Colorado potato beetle, black beet aphid, Apis faba, root knot nematode, pale cyst nematode, CLSO, the zebra which is affected by the tomato potato psyllid, and a range of exotic strains, biotypes, etc., of established pests and diseases. So tomato spindle tiroviroid, Aphis cipii resistant biotypes, other things like that. So I'll get to the first one. As Alan mentioned before, this year I'm based up in Cairns in the north with the federal government up here. So a lot of my focus, given the lockdown, has been on the situation in the north and in Queensland. But through the work that I've been doing up in Bowen area region and the Atherton regions, we've seen that the fall armyworm has had a significant impact. But I'd like to draw your attention to the fact that although fall armyworm first arrived in late 2019, early 2020, it was targeting sweet corn and sorghum and has had a devastating effect on those commodities. What we're now seeing is that those the focus and the populations are now spreading into more cash crops. So what we're seeing in the down in southern Queensland is to moving to some Asian vegetables, wombok and so forth. What we're seeing up in Bowen and Eyre is a move into chilies and capsicum crops. And what we're seeing up in Atherton is a move into the potato industry. So this is something to be aware of and be rather concerned about, because as you might all be aware, the fall armyworm can travel incredibly long distances. So even though its populations are stable year-round up in the north, you get migratory flights. And unfortunately this year, even those growers down in Tasmania were able to see that, that they were not out of reach of the psyllid having arrived there. Sorry, the fall armyworm having arrived there. Okay, but we are seeing some positives with beneficial control. Trichogramma is having some impact in the NT, but not so much in Queensland. We're finding beneficial insects being generous predators. Now, I'm just going to switch over to the leaf miners. This has been a real problem this year, and I would like to draw attention to 3 different leaf miners that look basically exactly the same. They are all members of the Trifolae family. We've had sativa, vegetable leaf miner, in the Torres Strait and contained up there for a number of years. Now, this has a potential to be a real risk if it gets out of the containment zones within that Queensland has established. Unfortunately, in October last year, we got the arrival of the American serpentine leaf miner, Lyromyza hudibrensis, in Queensland and New South Wales, arriving unfortunately through cut flowers, which is a major pathway of new pests and diseases. It was unable to be eradicated and is now a problem for both New South Wales and Queensland growers. Finally, unfortunately, as of last week, the American serpentine leaf miner, Lyromyza trifoli, has also been discovered in Australia. Now that's in Torres Strait and Kununurra. These all are incredibly resistant to some classes of chemical insecticide. Are able to resist a whole range of others because of the nature of their mining activity. And you can see that on the bottom there, that's some feeding damage from trifolae on potato crops in Africa. These things are a significant pest, and unfortunately, from what we're seeing in Queensland, their impact is spreading. They're not great flyers, but growing. So containment activities are probably our best bet, because once they're in the region, they're incredibly hard to control and manage. Finally, I'm conscious of time, we've got tomato potato. So it was a fantastic presentation from Sally there. Just an update, basically what we're seeing in West Australia is that there is no evidence from trapping and testing that CLSO is present. Yet again, from across all testing done over there, sampling and trapping has been conducted on all states and all regions around Australia, and has only been confined to West Australia. So this is great, good news. And then finally, that we're also seeing that the trap numbers in Perth and Kununurra, in Perth are dropping. So what you're seeing is the level of impact is decreasing. It's having a major effect on tomato and capsicum chili growers, but not so much on potato production. So that's great. Finally, in terms of the communique on the terminal movement agreement, is currently with the Plant Health Committee awaiting approval. So we have approval on the limit, no restrictions on potatoes for consumption, but with seed potato, that is still with the state and federal So that's it for me, and I appreciate you taking the time to listen. Thanks.
Thank you so much, Callum. All right, we have a bit of time for questions for Shakira and Callum, and thank you so much, Shakira and Callum, for those presentations. Really appreciate it. We've already had some good chatting in the chat box, and I see Shakira has been answering those as we go. Does anyone have any further questions for Shakira or Callum?
Sorry, I've got a question, Shakira. It looks like a great research project. So we're all keen to have commercial sentinels everywhere. So how is that looking?
Well, we are likely to start looking at, so what we need to do is we have a couple of benefit cost analyses on the go. So one is nearly finalised for grains, and we're about to commence one for horticulture, which I requested we do on potatoes. So that's part of the reason why I really want to focus on the Virginia trial that we've got coming up, so that we can really understand the benefit to industry, and then that will help us as we move into talking about commercialisation. So Hort Innovation are interested in commercialising the product, but we just need to have a look at its benefits. What we do have coming up that might be more adaptable, more cost-effective for industry is, instead of having these massive units with all the bits and bobs on them, we're looking at having individual boxes. So an individual spore sampler, they're much cheaper to produce, much easier to make a lot of them and disperse across the landscape. And so also looking at technology out of the BioScout startup project, which is looking at real-time identification of those airborne spores that it's collecting. So sort of buddying up with other surveillance tech.
So do you think, Shakira, that there's a room for citizen science, especially with the peri-urban issues around biosecurity? To, I guess, for insects, it will be difficult for diseases. I know that's done in ecology and environment and management.
I think so. I guess it all comes down to the diagnostics. You can have all sorts of surveillance tools, but you really need to have sharp diagnostic tools and having the right education out there, the right awareness. We're seeing a lot of interest from education providers in these sentinels. So maybe that level of citizen science, getting students involved.
Yeah, it could be. I mean, they're usually more interested in critters and insects than once we grow up. We're too busy. Thanks for that. Are there any other questions? Don't think I've seen any more.
Yeah, I've got a question here. This is Audrey from Simplot Australia. Hi, Shakira. I've got a question for you with regards to your spore trapping. Have you done any work to differentiate between what comes out from spores that are, stubble-borne that have been irrigated before, or are those basically from crop that are from a dry land mostly, so it's not irrigated per se? And do you see any differences between them in terms of spore dispersal?
We haven't really looked, I guess, because so far we've been looking into the function and operation of the sentinels themselves. As you can see from some of those images, we've really had this evolution of the engineering of the boxes. So we've really been focused on that side of things, and now we're starting to look at the landscapes and the environments we're deploying into. We have a deployment, the one for Griffith that is coming up, that is going to be at an irrigation trial site. So that's when we can start looking at those dry land versus irrigation and so on and so forth.
Right, okay, thank you.
All right, we might finish it there. Thank you very much, Shakira and Callum, for those presentations and answering questions. So our next speaker is Tim Belleville from Muir Sons. Tim, I see you right in the middle of my screen. Would you like to share your screen?
Certainly, thanks.
So Tim will be providing an overview of new crop protection products for the potato industry.
There we go. That's come up all right, is it, Kristen?
Yeah, you might just want to speak up a bit, Tim. You're a bit quiet.
Sure, no worries. Thank you. Yeah, thanks, Kristen. Thanks for inviting me to speak today. Thought I'd just do a quick overview for those who aren't aware. Most people are probably pretty much aware of Muir Sons, a reseller, of ag chem, fertilizer, and auxiliary products to the agricultural industry and certainly the potato industry. The company, certainly has grown. We're into our 90 years of operation. We're one of the first companies to have a national footprint with now 45 locations around Australia. So we probably cover off, I think, most of the major potato growing regions in Australia, and most would recognise the brand. And in Tasmania, our brand is AUSVEG. So yeah, I just asked to give a bit of an overview of the crop protection space and products that are coming. So I thought I'd just go over sort of three main points today. One is just the supplier industry that we obviously source products from that are available to growers, and growers would be familiar with and use. A little bit on the trends that I've seen occurring. I've been in the industry for 21 years, and yeah, just thought I'd give a little oversight on where I see the trends are heading for crop protection, and then just a few moments on some of the new products that are hitting the space that are relevant to potatoes in the current year. So we've sort of seen, this slide's a little bit of an older slide, but sort of back in the sort of around 2017 or since then, we've seen a real consolidation in our supplier base, in the base of companies that supply us, that you'll see in the marketplace, and we've seen that consolidation. And with that, I think really that's been a positive in many respects, because it has meant that the core R&D companies that we represent and that you will see in the market now are gaining a size that's allowing them to continue to invest in new products, and we're seeing new products come to the fold quite regularly. So it's a bit of an interesting take that we've gone from 10 to a dozen maybe major suppliers, and we're back to probably they've consolidated down, but I don't see that as a threat by any means. In many respects, I see it as a positive for product availability. So I thought I'd spend a little bit of time on trends and what's happening, in the space around products that are becoming available. Obviously, one of the key trends that we're always continually faced with is regulatory pressure. If you look at the arsenal of crop protection product that people have available to them, we have a real spread, I guess, of some very old chemistry to some newer chemistry. And then we're moving into a biological space as well. But we are seeing that older chemistry and perhaps even some of the what I'd call middle-aged chemistry is certainly becoming under regulatory pressure. And what we often see occurring in Australia is that we look at overseas and what some of the regulations are overseas, what the EU are looking at, and a lot of this older chemistry is certainly heavily under review. It's worker safety, food safety, environmental safety, all those aspects come into it, and as a result, we're starting to see some of that older chemistry fall out of our available products that we can use. I thought this slide was quite interesting. This was taken from a study that was from the European parliamentary research that was done back in 2019, and the graphs may be a little bit hard to read, but basically it's giving a snapshot of the amount of new chemistry that's been introduced into the market over time. and you can probably see towards the on the graph over here to the right, if you can see my cursor, as we've entered towards 2020, that there's been a fair drop off in the orange line is the amount of what we'd sort of call conventional chemistry that's coming to market. The amount of conventional chemistry that's coming to market is certainly tapering off. It's a lot less than what it was back in the 80s and 90s. And the blue bars represent the amount of biological chemistry that's coming to market. And we're seeing that is increasing. And as I said, with some of these bigger R&D companies getting bigger, they've got a bigger portfolio behind them, and they're investing heavily in new biological trends. In terms of, if we talk about biological products and the segments, what are they? There's the term biological gets thrown around quite a lot, and I picked this slide up a year or so ago, where I felt it sort of clearly put into bubbles the different areas of biologicals that we can look at, whether they're biofertilizers or biostimulants. They may be macroorganisms, and that's predatory insects and the like. And I see that sort of fits in with some of the earlier presentations today. But from a chemical point of view, there's a real concentration around what I would sort of term biochemicals, and then microbials as well. And the biochemicals is probably some of the products that we'd sort of term traditional crop protection, but they're coming now from a more organic background, and then we're also seeing lots of exploration going on around beneficial, whether it be bacteria or fungi, for use in the crop protection space. With that sort of background, I guess, in mind, I thought it was to have a look at a few of the newer products that have just hit the market or been in the market in the last year or two, and a couple of examples, I guess, of that biological space that we've seen introduced of recent times, and with a high degree of success, products that seem to be performing extremely well, even though they're, in the past we may have looked more cautiously on the biologicals, but I think our reliance on them is going to become higher, and we do need to look at how we can fit them into our programs. But EndoPrime, which is a product of offering from the Sumitomo group, is a product that, and you'll probably hear a bit more about it tomorrow in soil health, but it's a mycorrhizal beneficial fungi that we've been seeing in the field and getting really good results. And similarly, Bayer have recent times released a product called Serenade Prime, which many of you will be familiar with, and I guess, to show that the continual investment, there'll be an upgrade, if you like, of that product coming. They're continuing to invest in that Serenade space with Serenade Soil Active coming along. That won't necessarily be commercially available this year, but you will start to see Bayer with some product out and about this year to start to look at as they continue to improve the biological space. On the conventional, little bit more back on the conventional side, I guess, probably within the last year or so, you will have seen Miravis introduced by Syngenta, part of the newer chemistry, the SDH high group 7 chemistry, which really is has become a really good product for target spot in potatoes. So that product is new to the offering. It's become quite a good product to allow growers to switch to a different group. But an extension of that, in this season, Miravis Prime has been launched as well, which is, as I said, it's an extension of the Miravis family, if you like. It contains the Group 7 component as well, which they've now teamed up with a Group 12 fungicide, Flutioxanil, which has also been available in the Australian market for some time in other crops, but with a co-formulation that's broadened the disease spectrum that Maravis has. And if you've, I guess if you've really got a, prone to a sclerotinia, that's probably one of the main pickups that this product will bring to the table. So if you're in a high sclerotinia infection region, or it's a disease of concern for you, Maravis Prime over Maravis may well be the go-to option for you. And obviously its application would happen around when the sclerotinia is most prevalent, or you've got your most favourable conditions, which is probably early in the piece, or then later in the piece. Another new product, which again released this year by New Farm, a product called Emishield, which is a new mode of action. It's a group 21, so from a resistance point of view, another product that you can swap around. Has good action on both powdery scab and pink rot. It's an in-furrow or in-row treatment at the time of planting. Probably one of the important things to remember with this product is that it is a protectant. It's not a systemic, or doesn't have any movement as such. So that application is really important, that you get the application correct, because it needs to be around and in the right spot for it to be effective. But quite a good product to potentially swap out if you've been using some older chemistry and struggling to get good control of either powdery scab or pink rot. Couple of small graphs, and I won't labour on those, but really just showing that it is showing good effectiveness against some of the other products that Medley and the like that New Farm have had in their stable, and just going to a newer and improved chemistry. New Farm also will be releasing a new formulation of difenconazole. Difenconazole is quite an old piece of chemistry that's been available for quite some time, but I guess they're going from an EC formulation to an EW formulation, so an emulsion in water, and one of the benefits that we'll recognise with that formulation will be worker safety, I guess, as much as anything, going from a hydrocarbon-based product to a product that doesn't contain that hydrocarbon base. So they'd receive feedback from growers in many industries that worker safety continues to be something that's important, and they've changed the formulation of that particular product. And just the last product, which is on its way. It's not hasn't actually finished APVMA approval yet, but everything going well. Bayer hope that they will get the tick off from the APVMA towards the end of this year. Is a new product called Xavanto Prime, and that'll be targeting whitefly and green peach aphid. Again, it's new chemistry, a group 4D. The bit of research I did, I couldn't see any other group 4D chemistry available at the moment. So that'll be a new offering. Again, a good rotation partner for pesticide resistance point of view. Again, I guess what we call more conventional crop protection, but again, you're seeing most of these new products that are coming certainly do have a soft angle to them. So it's something that, obviously before it's released, there's plenty of testing done around compatibility with IPM programs and safety to bees and other beneficials. So they were just a few of the products that I thought I would highlight today, and I think, Kristen, I've probably just about reached the end of my time, if not gone over. So thank you. And if there's any questions or comments.
Thank you so much, Tim. That was really interesting. Also, love to see some of that earlier data about some of the trends regarding the type of products, and I guess the increase in biologicals and the gradual decrease of some of the more conventional chemistry. please type in or put your hand up if you've got some. Oh, here we've got a question come through. I guess I had a particular question just in regards to, do you see those trends continuing on? And it's great. And I guess the other aspect of that is it's great to see some different group chemistry coming in to kind of to try and combat some of those resistance issues. Do you see that continuing as well?
Yeah, I guess the new groups, I mean, all companies are obviously are looking for new groups and new chemistry. Yeah, resistance is probably top of everybody's mind. So that is certainly something that we're seeing. But I think as we seem to be exhausting the chemistry that is available. So yeah, the new groups seem to be getting harder to find, and they're coming through, I would say, a little more slowly, and some of those graphs probably depict that you know, they are coming, but they're coming a little more slowly. I think there was a stat there that, you know, takes I think it was taking somewhere around 8 years back, perhaps in the late 90s, early 2000s, to, you know, from discovery of a new active to get it to market. And now I think that's probably out to, there's a number of industry people on today who may correct me, but I think that would be out to somewhere around 13 to 15 plus years now, just with the, you know, there's a lot more regulatory involvement, there's a lot more research that's done. So yeah, as the newer chemistry does get to market, I think we can be quite assured that, you know, the right safety profile's been done behind it, but it does mean that things are a little slower in that conventional space. And the biological piece, yeah, certainly there's been a lot of investment, and that's probably in many respects can sometimes be a little bit slower, even more than conventional, because we need to fully understand how the product works. It may be a little more unpredictable than what conventional chemistry is, so you need to get a really good hand. And there's lots of environmental factors that you need to take into account that can affect how a biological product may perform.
Tim, do you know of any biologicals in the pipeline for registration? We know there's several hundred products floating about in that, you know, wider biologicals area, but we don't see many registrations.
Yeah, that is true. I mean, as you say, I know of lots that are getting trialled and not too far away potentially, but it does take a long time to get the registration, and I'd be hesitant to. We're often hesitant to say, you know, there's one not too far away until it has all the approvals, because then can be disappointing that it didn't make it all the way to the end. Yeah.
Yeah, I guess we've got the hurdle in Australia that we have to follow the same process as synthetic insecticides, the same APVMA approval process, whereas in the US and the EU there is a different process. So not sure whether we ever get a different process that actually allows, you know, higher rate of registration, rather than having all these weird and wonderful products. With, you know, Jenny had the question, how do we know in a biological product which contains a beneficial microorganism, whether it's actually alive or dead?
Sorry, I missed what was the exact.
Oh, the question is, you know, for instance, in ceranide, how can we,
Yeah.
or other products, how can we be confident that the organism actually is alive?
Yeah, well, I guess that's, you know, I mean, when you buy any product, I would say if it's a research product from a proper R&D company, then we can be confident that is the case. It doesn't get to market unless they can guarantee that. And I suppose that's a relevant point, that with a lot of the biologicals, you will see an expiration date on the product. So often those products do only have a 12-month or maybe 2-year shelf life. So yeah, when we're switching to those products, you need to be mindful that you want to be getting nice, fresh product. It also produces challenges around production of those products, because they need to be obviously cultured and produced. And if we get a large a trend of, you know, adoption quite quickly, then sometimes production can't keep up with the adoption demands. Yeah.
Yeah, and there's probably one comment about these type of products that don't work in the same way as synthetic pesticides. So if soil health, general production management is so much more important to get the added benefits of biologicals. So they're not just out there killing pathogens.
No, that's right, and yeah, it's I guess in many respects, the change over to a biological is a lot more complicated, and it requires a bit of a broader scope of thinking rather than just see a pest, target a pest, and spray a pest, I guess.
There's also actually a good question there about would it be better to actually foster biologicals, microorganisms, a healthy growing environment rather than trying to add them to the system? What's your view on that?
I think it's a combination approach, actually. Yeah, I think you need to the fostering and the cultural practices are always extremely important, even with when we've been in the past using conventional chemistry. Often if your cultural practices and the like aren't up to scratch, you're asking far too much of a product to do all the lifting on its own, and therefore a combination of both and a prevention is always much better than cure. Yeah.
Yep. So I think that might be there's some comments, but now further questions.
Yeah, thanks, Tim, for that. That was really, really interesting. So we're going to move to the panel session for the forum, which will run for the next 30 minutes. And really, this is an opportunity to ask questions of any of the speakers that we've had today. I'd also like to introduce Dr. Paul Horn. Paul, do you want to turn your screen on? There it is. I'd like to introduce Paul Horn from IPM Technologies, who I asked to join the panel session today. So Paul is the Director of IPM Technologies, and alongside Jessica Page provides advice to growers on managing pests on their farm using an integrated management approach. And Paul and Jessie have also been delivering the IPM program for the potato industry across Australia, which has involved on-farm demonstration of how IPM can work and does work. So I thought Paul would round out the panel nicely and can answer any IPM related questions. So the focus of the panel is on how new technology and products will help us manage pests and soil health into the future. So we've heard today from a range of speakers on some of the new pests or new biological products such as Tamarixia, which might help us manage pests, and also some technology that's been around for a while, I suppose, EM38 soil mapping. But wanted to open up the floor, and we might be able to do this if you want to either type in a question into the chat function. Doris and I can direct those questions, or if you want to put your hand up or ask it in person, you can do that too. So I might hand it over to you, Doris.
Yeah, so you can put your hand up just by putting your hand up, but there's also a little reactions icon on your bottom of your screen, and you could put your hand up, your electronic hand, if you don't want to hold your own hand up forever. I might actually just ask Paul briefly could you say a few sentences about the IPM program, so people who haven't heard about it can just get a bit of an overview, and also what, from your work, just what you think is the greatest opportunity for the potato industry as far as IPM is concerned.
Hey, thanks, Doris. For the last 4.5 years, we've had a project that is funded by the potato and onion industries and Hort Innovation to basically show how IPM works to anyone that's interested, any potato grower, any advisor around the country. So over that period, so the project comes to an end in December, and the last year has been terribly disrupted because we haven't been able to go hardly anywhere. But where we have, we've reached pretty much all of the major potato growing districts around the country, and we've demonstrated IPM and offered support to those people that are take it on. And I know, I'm looking at some of the names on your meeting, I think quite a few people have taken it up. I think I spoke to Kristen about whether I should talk at this meeting, and I said no. I think everyone's heard me so many times it would just put people off. So I don't think I need to say too much more.
But maybe a great opportunity or a great challenge from your point of view, because I know from many producers that taking on IPM is a bit nerve-wracking, because you have this, not habit, but you need to spray, and suddenly somebody tells you don't have to. What's just from the experience, what would be the way to start?
Yeah, so that is the whole purpose of the project, where we, especially dealing with advisors, we take that risk away. So we give the advice. And so then both the farmer and the advisor can see IPM in action. And usually there's a reduction in pesticide use. It doesn't have to be an elimination of all insecticides, but certainly there's typically a massive reduction if people have been relying on it. So yeah, there's an opportunity, and I have to say, from our point of view, it's been very successful. We haven't convinced every potato grower in the country to use IPM, but I would have to say the majority now do, in my
That's
opinion.
right. Yeah, thanks. Is there actually any IPM for psyllid in Australia, or are we still working on that for the TPP?
Yeah, so actually we had a project funded by the potato industry before it even got here to work with Plant and Food in New Zealand, and we developed an approach there. And if it arrives here, I think we're ready. There's a range of beneficials that we know for sure will eat it. Its occurrence in Western Australia has been of concern, not to potato growers directly, apart from the barriers to trade. And I believe it's because we have enough natural enemies that can currently take care of it. So we don't have specific parasitoids like Tamarixia, but we have generalist predators. And if farmers are using an IPM approach, they're already using the things that will eat potato psyllid. So they're damsel bugs, lacewings, hoverflies, they will all eat tomato potato psyllid.
So there's a real call for using IPM to actually be prepared to have the predators there.
Absolutely, yeah.
Maybe also a quick question to Callum about, people are very frightened of the trade restrictions. I think you might have some good news there.
Look, I touched on that, but we presented to Plant Health Committee yesterday, which is the body made up of the federal and the chief plant health managers for each state. They're taking under advisement, and they will be back with a ruling or an answer on that seed potato movement soon. But like I said, I hope we've publicised this sufficiently over the previous 6 months or so. But there was a communique reached with the help of Alan Smith and a whole bunch of other people, McCain's and elsewhere, to basically nut out a solution to the risk posed by psyllid moving on potatoes for human consumption, and allaying the fears of the regulatory bodies in the eastern states, which were founded, but were allayed. And so now, if there is a detection of psyllid in the east, then potatoes for processing will be able to move. Potatoes for consumption will be able to move across state. So yeah, that is a positive, but still the wheels of government take time, and it's just a little bit frustrating that taken such a long time to get where we are to now.
Thanks, Callum. Are there any further questions to the panel? Remember, on Precision Ag, Shakira for the IMAP, or Callum or Paul who joined us. It's your chance. Also Tim, if there's more questions to Tim.
Greg, I was wondering if I could ask a question in regards to some of the precision ag development. So you've obviously really using the soil EM38 soil mapping to really direct how you apply nutrients and manage your soil. Do you see a time when perhaps it's also possible to map, you know, pest hotspots or disease hotspots and then try and tie that in with some of that soil mapping to see if there's a correlation? Have you seen that used at all?
Yeah, I think so. And we've already had a couple of people this year who have done some mapping comment around certain areas of the field that were perhaps low in nutrient being perhaps, you know, where a prevalence of a certain disease or insect was found. Now, whether that's because it was a soil type thing and therefore the soil type was the factor making the nutrient levels low, or the soil type in some other way was perhaps affecting you know, disease or insect use, I'm not really sure. But I'm certainly interested tomorrow to listen to some of the stuff on pink rot and just some of the other presentations that we're going to see tomorrow around, certainly around disease mapping, because I think there's a lot that we're going to learn, and we have really focused on just, you know, P, K and pH this year really with some of the production issues. But I think the additional information we see on these reports we don't really yet understand or we don't know about, so it's what we'll see going forward. And it's quite interesting talking to, you know, farmers and agros when they look through some of these maps. They start going back over the years and going, I remember, you know, 3 or 4 years ago having a particular problem in this crop, in this corner or in this part of the paddock, and perhaps it's related to this nutrient or, you know, pH or what have you. So I think, you know, we're still really at the start of what we are going to understand and what's going to unravel it as we get further into it.
Mm. And that was really one thought I had, thinking how we're putting it all together into a production system approach, because for the plant or the farm or the agronomist, everything happens at once, you know, and everything is related. So whereas in research, we work very much in silos. So it'd be really, I think, interesting to put more knowledge together, like the mapping and looking, is there higher disease incidence of, for instance, pink rot when the pH is lower, or something like that would be, I think, really valuable.
The growers I've spoken to are quite open to that. You know, they want to understand whether there's a correlation or not, and they're time poor, a bit like everyone. So if someone else can sort of look at it and understand it, I think they're quite keen to share that information and understand it better.
Yeah, so there's a research idea. Tim, have you monitored the question? There's one for you on chlorothalonil. You can look in the crystal ball, maybe.
Yeah. Difficult to answer, I guess. You know, with certainty what the end result may be. But I would like to think that, you know, the Australian regulators and the APVMA in particular do take quite a science-based approach to their decisions around the allowance or non-allowance of chemistry. And generally speaking, I think with most industries, you know, obviously safety is paramount, but often it'll also be looked as, you know, other viable alternatives that are available for a cropping segment. I'm not sure that it's too often that we see a product pulled that leaves us with no other alternative. So that probably doesn't answer the question with certainty, but I think it's available for the foreseeable future. What that looks like down the track, you know, there's lots of variables that may or may not take place. But we have seen some other crops, not necessarily potatoes, but other crops that use the product that have probably changed their use pattern a little bit. They might have used it later in the crop cycle and they've started to bring it forward earlier into the crop cycle so that they can get away from or reduce their risk of seeing residues in the end produce. So that wasn't something I had in my presentation, but that is certainly something, you know, from a consumer end, we're obviously seeing this this drive for low residue or nil residue, and that's something that's also becoming more important and obviously being closely looked at in the development and usage of the chemistry.
Yep, so we will actually have a soil wealth webinar on the 25th of August looking at what's happening or has been happening in Europe on mandatory IPM, which is IPM in Europe is actually pests and diseases, and look at what does it mean for Australia. So I'm sure you can keep your eyes peeled for that if you're interested in listening to. We're trying to get some speakers from Europe who have actually experienced things firsthand. And the other thing is, I mean, Europe is a bit different, obviously, because the population density is very high and contamination has been so much more severe. So we have a bit of an advantage. On the other hand, you know, it's really worthwhile looking at what's happening there. I actually had sorry?
No, you're right, you go.
Oh, a question back to nutrition and precision ag is the real-time nutrient monitoring. I know about the nitrogen, you know, color-based monitoring, but I don't know whether there's any other in-field nutrient monitoring apart from some probes. Actually, I know that Swan Systems and others have again probes that look at nitrogen and potassium. But is there anything else, Ben?
All great.
Oh, great.
Yes. Certainly it's a space that's developing, but you know, outside, I guess some fairly rudimentary measurements with regards to probes and bits and pieces, it's still probably too early for it to be considered accurate enough. So, you know, the ideal situation would be just to roll through a paddock and punch a probe in the ground and just to get a reading as you're going. That'd be great. But we certainly haven't seen anything that's as reliable yet as just, you know, a soil test or wet chemistry, if you like, which obviously there's a cost to that, but it doesn't feel to me like that's too far away. And certainly, you know, you'd like to think within the next 10 years or so, there'll be certainly technology that will exist that will allow us to do that.
Yeah, I guess what we've seen with the, you know, the GRDC video, you wonder whether there's something that could be actually attached to a tractor and monitor any risks as you go across the paddock. That sounds not bad, I guess.
Yeah, it's sort of like the stuff of science fiction, isn't it? But just, I was thinking the same thing watching that video. How good would that be to just have a probe, a remote probe that just sat in your field full time and gave you real, you know, readings all the time? I mean, that would be fantastic.
There's actually also one question that came to mind just because yesterday we ran a seminar on herbicide resistance, especially ryegrass. And we seem to be talking about pests and diseases a lot, but not weeds. And I wonder whether anybody has anything to offer from the panel or even from the audience on how we're going with weed control in potatoes. Is there any challenges, any issues? I mean, the ryegrass, herbicide-resistant ryegrass certainly looks like a huge problem, but we don't seem to talk about it in horticulture. Any comments, Tim, maybe?
Yeah, it's a space that we, as you say, in potatoes, we don't see a lot of work being done on new herbicides. I'm aware of one that is in a trial stage. All going well. You know, a couple of years away, but it's something that I guess the herbicides, the broadacre crops tend to get the focus with the herbicides coming through, and probably horticulture in general, we don't see as much focus on herbicides. It's probably there's lots of variables, there's lots of off target and potential replant and plant back issues that come with using herbicides in horticulture, and therefore it's probably a longer process to bring a new product market with surety. So I think that's why we don't see them coming as often. Yeah.
We certainly had a huge discussion yesterday on alternative methods. So if anybody thinks we should initiate research in the potato industry on different herbicide or actually weed management, please let us know. I think there has been work done in vegetables, not much potato specific. We know there's microwave technology coming around, but again, it's in development. It's slow at the moment, but reasonably effective also against soil-borne diseases. But if there's a research need, especially also looking at resistance, ryegrass, white radish, it would be good to know before it becomes a real big problem.
Sally, I had a question for you, and this might be a bit outside of the sphere of your project, but how are growers in New Zealand going with managing TPP now that they're several years down the track?
Yeah, thanks, Kirsten. I think that they're managing. There's a lot more, as I said, there's a lot more chemistry in the toolbox for growers now. So potatoes obviously got quite a few registrations for products like cyanotraniliprole and things, but they're also, as Paul alluded to, they're also a lot more smarter about monitoring their crops and having crop scouts through to actually measure and monitor for psyllid. So they're actually making more informed decisions around spraying and not just doing those calendar sprays. They're actually responding to pest and disease pressure. So I think that's been a real learning from the industry. I guess what we need to do now that we have Tamarixia and it's slowly starting to establish itself in New Zealand, we still need to encourage growers to release. I see that in the chat there's been quite a few comments around beneficials in Australia and possible use of beneficial insects for controlling psyllid in Australia. And yes, we did look at that. We've got a similar suite of ladybirds and lacewings and hoverflies here in New Zealand. And what we found is that they're not specific to psyllid. Yes, they will control to a certain extent, but then when psyllid actually gets away in a crop, those other beneficial predators really get outcompeted. And that's where Tamarixia comes in, as that extra tool and that biological control toolkit. So we're still learning how to use Tamarixia, and with more monitoring and more research, hopefully, particularly from Plant and Food, we'll hopefully be able to transfer that information across to Australian potato growers.
Thanks, Sally. Paul, I was just wondering, from your experience and insight doing the IPM and potatoes project over the last few years, how have you found the level of uptake of using an IPM approach? And I guess perhaps some thoughts on how we can encourage people to use that sort of approach in the potato industry.
I think that there's been really positive uptake. Like I said, with this project, we can't force people to work with us or use IPM. No one can. But there's been many major companies that have adopted it, as well as individual small farmers. But I think one of the biggest successes has been to get reseller agronomists and advisors to understand IPM and see that it's not so scary. And that's been the way to actually get it adopted, because there isn't a crisis in pest management in Australia in potatoes. And unfortunately, the experience is that people keep doing the same thing until there's a crisis and they're forced to change. That hasn't happened. So to get a very high level of adoption in the absence of a crisis is a real success. So the information's out there. If there ever becomes a crisis, and that could be simply the withdrawal of chemicals that people are used to using, then that'll be for the future. One of the most difficult things to get across is with IPM, the I stands for integration. integrated pest management and to integrate pesticide use with beneficials. So there's a lot of information that people need about what is a soft product and what is not. And these days, there's so many new products on the market that are safe to some beneficials, even a lot of beneficials, but not all beneficials. So if they're marketed as IPM compatible or safe products, they almost certainly are in some crops, but not others. And so having grower and advisors understand which products can be used safely and which can't, that's the key in a lot of cases to successful IPM.
Thank you very much, Paul.
No worries.
We've had a question, is there potential for an industry platform similar to the list map for ag spatial data to bring it all to one usable location with grower IP protection?
Just for the non-Tasmanian list is Land Information Systems Tasmania. So that means spatial information. I think every state has got their own system. But you know, at the moment we can look up land use and soils and land capability and where the boat ramps are and risks of waterlogging and salinity. It's a lot of information actually there, and a lot of farmers and landholders actually don't know how much information is there. However, for instance, talking about, even yesterday we thought we could get the audience to map herbicide resistance in Tasmanian paddocks. That wasn't taken up very nice because there's a lot of competition, there's a lot of IP, there's a lot of industry angst around these things. But in principle, it's a good idea, but it would be interesting to hear comments from others. So now, I believe that even we've been talking about, you know, some are recording monitoring data, which you know, for biosecurity purposes, and that certainly has been a difficult discussion of how can we de-identify locations and you know, make the granularity higher so you cannot identify individual properties. I don't think anybody would really like that, to have a pin on their paddock saying, you know, we found whatever disease here last year. Wouldn't go down too well. So we need, I guess, to think about how far a mapping information system could go without making it, you know, so to protect the businesses as well.
Right. Well, we might, unless there's any final questions for our panel. Just checking the screens. We might move on to the next section. Doris, do you want to just explain a bit about?
Oh, yes. So we thought maybe some of you would like to stay on and talk on you know, breakout session. So we will put every speaker into their own little room, virtual room, and you can actually self-select whether you want to have a more, you know, smaller scale communication with any of the presenters. So Kristen will bring up the list of presenters, and you can actually, once it's up and you see the list, you can actually click beside the name and you will be zoomed, like in, you know, magic. You'll be zoomed into that discussion room. So it's not, the beam is nearly there. And the way you actually can do that, you have to hover over, there's a number beside the name of that person, so you can hover over that number once it appears and be transmitted to that room.